Thanks, Jack. I really appreciate the introduction, and I'd like to thank Technology Networks for giving me this opportunity to share this data with you guys. Again, my talk is going to be entitled Single-Digit ppm Level of Host Cell Proteins Using a Simple 1D Data-Independent LC-MS Acquisition Method Utilizing a CHO Spectral Library. So why would we want to perform host cell protein analysis? Biological therapeutics are produced in cells. CHO cells produce 7,000 plus proteins alongside your protein of interest. And host cell proteins are a mixture of unknown proteins with uncertain impacts to product quality and patient immunogenicity. Therefore, they must be tightly controlled. Typical host cell protein levels at different stages in the purification process can vary from anywhere to 500,000 to 1,000,000 parts per million, or nanograms of host cell protein per milligram of antibody. After the protein A stage, you will typically have 5 to 50,000 nanograms per milligram. After cation exchange, it should drop down about 10 to 100, and after anion exchange, it should be less than 1 to 10. A typical specification is somewhere between 1 and 100 parts per million or nanograms per milligram of antibody. And of course, the specification depends on the nature of the host cell protein, your process history, patient population, dose, and a few other factors. Typically, ELISA is used to assay for host cell proteins, but as you can see on this 2D Western blot and silver stain overlay, typical ELISAs provide 50 to 80% coverage of the total host cell protein content. Now, I should say this was done by overlaying the Western blot generated by the ELISA antibody from a sort of standard off-the-shelf host cell protein kit, and then we compared that with the silver stain and found that there's a lot of places that are being missed by the ELISA antibody. However, this is the current standard ELISA assay to detect host cell proteins. Polyclonal serum is usually raised against the host cell proteome in rabbits or goats, and then you produce an ELISA detection reagent. It can be time-consuming. You have to inoculate the animals, wait for their titers to come up, purify out the antibody, and then test it. And then it can be very difficult to obtain 100% coverage of the secretome. We are working on adopting mass spectrometry as an orthogonal method that is less biased than for host cell protein analysis. So which host cell proteins can humans tolerate and which would harm human life? This is really the big question. And one of the things that we rely on is the immune system of our host organism to generate an ELISA reagent. However, the immunogenicity towards these host cell proteins varies by species. Serum raised in rabbits and goats may fail to detect a host cell protein that is highly immunogenic in humans. Any protein with greater than 98% homology between CHO and rabbit will not generate a robust immune response. And the picture here just illustrates that rabbits are not equal to people. Okay. At StemCentrix, we are still developing an ELISA reagent, but we're not using just one organism. We're working on developing a reagent in chickens, rabbits, and goats, and we're basically doing everything we can to ensure a robust ELISA assay with good coverage. But we're also